Hello, this is Mr. Wertz. Welcome to Chapter 2, Section 1, Viruses. Please remember to have out your green fill-in-the-blank notes sheet and fill in the blanks as we go throughout the notes. Here we go. Viruses are microscopic particles made up of a DNA or RNA center covered by a protein coat. They are not classified into any kingdom of organisms, so they're not really classified as animals, plants, bacteria, fungi kingdom, or the protista kingdom. They show almost no characteristics of living things. Basically, about the only similarity they have with living things is that they reproduce. Viruses are classified by their shape, number one. Number two, they're classified by the kind of nucleic acid it contains, the DNA or RNA, which basically is the instructions the virus has when spreading. And then finally, number three, the kind of organism it infects. Does it infect a human or a person? Does it infect a different animal like a cat or a dog or a horse or a cow? Or maybe a plant. Maybe it's a certain type of corn or a certain species of tree that it infects. So these are the way that scientists group or classify viruses. Viruses are considered to be parasites. A parasite depends on another organism or cell to survive and may harm that organism or cell. So they live off another organism, and that other organism is called the host, the cell or organism that the virus depends on. You can see in the picture here, these weird-shaped viruses are invading this cell, making copies, reproducing, and then spreading. The parasite, the virus, is living off the host cell. Viruses can be active or passive. An active virus causes the cell to make new viruses and destroys that host cell. And there's 4 steps in this process. First of all, the virus attaches to the cell's surface. It invades, which means the nucleic acid, or the instructions of the virus, inject itself into the cell. Copy. The new virus particles are made. They make copies of the original. And then they expand and expand and expand until the cell can hold them no longer, and they burst and release. The cell is destroyed as new viruses are released, and they go on to other cells and continue this process over and over and over again. That's an active virus. 4 steps on how they function. The other type is a latent virus, which is a virus that enters a cell and becomes part of the cell's DNA without destroying the cell or making new copies of the virus. It's important to realize, you guys, that latent viruses can become active at any time. For example, many of you have had cold sores or canker sores before. You don't always have those, but they flare up or become active at certain times. That is an example of a latent virus. You have that in your body at all times. It just flares up. I always remember the term late. It could occur later in its lifetime. Let's look at a vaccine, often called a shot. A vaccine is a substance made from damaged virus particles that cannot cause disease anymore. Remember when your parents said, "Oh, it's just a little poke"? Well, that's usually not a little poke. But, oops, let's go back just a second. There we go. It's important to realize that once you have a virus, you can't really get rid of it. There's no medications that can cure viral diseases, but some can be prevented by vaccines. For example, a lot of us get the flu shot to prevent the flu virus from getting into your body. You might have had the chickenpox vaccination. You might have had a hepatitis B vaccination. Maybe you had smallpox, mumps, measles. There's all kinds of vaccinations that kids get growing up that help prevent viruses from getting into your body and spreading. The very first vaccine was for the virus smallpox. It was developed by a scientist named Edward Jenner. And Edward, there's a picture there, grew up in a rural area, kind of a farming community outside of London, England. And he noticed that a lot of the people in his community were dying from smallpox, thousands of people. It was a serious problem. But he noticed that there were some other people in his community that actually never got smallpox. And he actually did some research on this, and he created the first vaccine from a totally different virus called cowpox. And we're going to watch a video on that in just a second. But basically, in a short way to explain this, it's kind of complicated. But the women in those days were called milkmaids. They stayed home on the farm while the farmers were out in the fields. And they milked the cows. That's why they're called milkmaids. Well, a lot of times, some cows would have these sores all over their udders where she's milking them, called cowpox. And those sores would spread to the women. Get in their hands, their wrists, and their arms, and they'd be kind of nasty looking pimples and blisters and just sores and everything that would sometimes bleed. And he realized that they did not get smallpox, the deadly virus. Cowpox wasn't deadly. So what he did, this is so bizarre, he actually took a sample of the women's cowpox, the milkmaids, and injected it into a boy in the community, really giving him cowpox. Then basically he tried to inject the smallpox virus into that boy, but the cowpox protected him from getting smallpox. And from there is how he developed the first vaccine.
Finally, nearly 30 years after first hearing about cowpox, Jenner decided to experiment. Now, all he needed was a milkmaid, and in May 1796, she walked through his surgery door. This is possibly the most famous medical image of all time. It shows the hand of Sarah Nelmes infected the pustules of cowpox. Jenner took an implement like this, possibly this very one, and with it removed the liquid from the sores and infected a young boy. His name was James Phipps. 8-year-old Phipps went on to develop cowpox as Jenner hoped. When the illness had passed, Jenner again infected him, this time with smallpox. 2 weeks later, Jenner felt able to write to a friend, "I have at length accomplished what I have been so long waiting for, the passing of the vaccine virus from one human being to another. Now, listen to the most delightful part of my story. The boy has since been inoculated for the smallpox, which, as I venture to predict, produced no effect."
Was Jenner really brilliant, or did he just stumble upon something by accident?
Well, I think he was both of those things. He did stumble across the discovery. Other people had already done that, so he was not the first to make the connection between cowpox and protection against smallpox. But the crucial thing that he did was to broadcast the news to the world. And he did that in a very imaginative way. In fact, the first paper he submitted was rejected by the Royal Society. So he then did a thing that I think would not be smiled upon these days. He actually had the paper self-published. And when it came out, it sold like hotcakes. And people read it, and they clearly thought, wow, this is something new.
Flawed but brilliant, Jenner's inquiry showed that vaccination with cowpox actually worked and saved lives. So why wasn't it immediately accepted?
Well, I think to understand this, we need to think about the world in which Jenner was publishing and working. Jenner is, after all, essentially a country doctor, and then as now, it's the London doctors, it's the metropolitan doctors who see themselves as being in charge of medicine. So what we see here is a constant battle, a kind of natural selection process between lots of different individual practitioners, trying to rubbish the discoveries, the methods, the techniques of other practitioners and support their own. And it's into this debate that Jenner's work steps.
So what was the reaction to the publication of Jenner's results?
Well, within this battle, there are really sort of three forces. There are people who oppose both Jenner as an individual, as a character, and his method of vaccination. There are then those who like vaccination as a method, but don't like Jenner as an individual. And finally, there are those, very often friends of Jenner, who like both him and his method. So very early on, the battle lines are drawn.
Other studies that scientists use viruses for. Interferon is a natural chemical produced in animal cells that stops viruses from infecting other cells within that animal. All animals naturally produce this, and basically I like the word interfere or stops. This chemical interferes with viruses getting into our bodies and stopping them from spreading. And what scientists are doing is trying to figure out how they can duplicate this or make a synthetic version of interferon so they can create like a vaccine that would work for everything. They're way off on that, on those results yet, but they're starting to look more at these animal interferons to see what they can do to make a generic virus or vaccine. Also, scientists use gene therapy, which is when viruses can be used to correct damaged DNA in specific cells. The virus actually is a carrier of DNA, and I'll explain that right here. Scientists will actually take healthy DNA from one person, mix it or inject it with a virus, and hoping that the healthy DNA will spread into the unhealthy person's body, distributing or dropping off that healthy DNA to correct the unhealthy DNA. Scientists, again, are still working on this to see the progress, but there's a lot of new research that I think is going to help us, by the time we're a lot older, or maybe our kids or grandkids, way years from now. So anyhow, that is our notes on viruses. Thanks, and see you tomorrow.